Hello everyone, this is Lecture 18 on the auditory and vestibular systems. So we're going to talk about the transduction of wave and of accelerational forces on the head. So first we're going to talk about the auditory system. So first, what is sound? Well, sound is a traveling wave of pressure. So we can image that here. So imagine each one of these little dots is an air molecule, and if some source like a gong or something starts to vibrate, it's going to cause a high density, which then propagates through space. And so we can represent that down here as the pressure. So here's high pressure followed by low pressure, and it's moving outwards. And there are four properties that are important. The first is the amplitude, that's how loud it is, and that's the difference between the high pressure and the low pressure. That corresponds to the peaks of the sinusoidal wave here. The frequency, that's how close together these are, and we perceive that as pitch. Phase information, which has to do with location, we'll talk about that a little bit later, and the waveform. So this can be more complicated, which provides information about the environment. So waves lose energy as they propagate outwards. And you can think about this as dropping a pebble into a pond. So right where the pebble goes in, the amplitude is quite high, but then as it spreads out, the energy is spread out over a larger and larger area, and so that the amplitude decreases with distance. But the auditory systems in humans, and especially other animals, is very highly attuned and very sensitive. So the threshold of human hearing corresponds to about a 1 watt source heard at about Edmonton. So if someone had a quiet car stereo, if there were no other sounds in the environment, that would be the threshold of detection. But of course, there's a lot of other sounds, and there's trees in the way and all that. So, practically you could never expect to do that. So what I showed before was just a pure sinusoidal wave. Sounds in nature are a lot more complex, especially speech. So this is an example of that tone, which would be something like that. But here is representation of the word saw. And this is the waveform that we would see. So if we had a microphone, we would see the s part of saw has a lot of low amplitude but pretty high frequency. By the time we get to the ah part, we can see that it's lower frequency but higher amplitude, and there's some other stuff in here. So it's hard to appreciate what's going on with just the traces in time, so traces of pressure in time. So instead, what we oftentimes do is look at a representation that is frequency, which is on the vertical axis, and in time, which is on the horizontal axis. And the amount of power or amplitude is now shown by how dark it is. So here, all the power is in this one frequency, which is at 5,000 hertz. And so here's a band that's right at 5,000 hertz. But when we have the word saw, you can see that this in saw has frequencies that are distributed, from maybe 4,000 up to 11,000. And then when we get to awe, we can see that there's a number of different bands at different frequencies, and these things change a little bit. And the reason different people's voices sound different is that these bands will be in slightly different places or more energy in some bands rather than other bands. These are referred to as spectrograms, and it's a very common way of analyzing things that change in time. Now, let's look at a couple other vowel sounds. So here's aw, which is kind of similar to what we just saw with the saw word. Aw has a number of bands in a number of different frequencies. E looks quite different. Notice, this part is completely missing in E. And here's ooh, which has, mostly low frequency stuff and less high frequency stuff. And these are referred to as formants. Okay, so how do we perceive this stuff? Well, the first step in the process is to go from pressure waves into a neural signal. So let's look at the ear. This is done in the ear. So this outer part is referred to as the pinna, and the job of this part is to reflect sound into the ear canal. This is the ear canal, which then funnels sounds in. It comes in and hits the tympanum or eardrum, which then vibrates. There are 3 bones, collectively referred to as ossicles, that are connected to this, the malleus, incus, and staples. And we'll see an expanded view here in a second. Here's the cochleus. This is where sounds are going to be transduced into a neural signal. And coming out of this is the cochlear nerve, which is going to have the neural impulses of sound, and that's going to go to the central nervous system. In the second part of this lecture, we're going to talk about these semicircular canals that you can see here, and this is for the vestibular sense, or the sense of head acceleration. Okay, so now we're going to zoom in. So here's the eardrum, the tympanum, here's the malleus, incus, and stapes, and these connect to a piece of tissue that's called the oval window. And the reason it's set up like this is that this is the cochleus, and normally it's kind of wound up like a snail, but here it's stretched out. And so we're going to see in most of the diagrams, we're going to see it kind of unwound and stretched out because it's a little bit easier to perceive that way. And this thing is filled with liquid. The reason it's filled with liquid is because this is the external solution for neural cells, and therefore it has to have ions, and the ions have to be in solution. So now we have this problem that we have to go from pressure waves in air to water. And so most of the time when you have pressure waves and you have materials that are very different density, so air to water, and water is much more dense, most of the sound energy will just reflect off. So this is why if you're underwater and someone's calling to you, you can't hear them very well because the sound is just reflecting off. And so the inner ear has a clever solution to this problem. So we don't want to reflect that energy, but instead we want to convert that into some signal inside the cochleus. So how are we going to do that? Well, the eardrum vibrates with the pressure, and this is fairly big, and this comes down to a small window via this mechanical linkage. And so what this is doing is this oval window, on this side is air, on this side is liquid, and so it mechanically pushes this up and down. And so, with the pool analogy, it would be like having some membrane on top of the pool, and then you're pushing up and down. Because the area of this is big, much bigger than this, we have mechanical amplification. And so most of the pressures, waves that are coming in are being transferred into the liquid. And we'll see what happens. Because liquid is non-compressible, as this presses in, there's another flexible part of this membrane called the round window that pops out. So when this pushes in, this pops out. When this pulls out, this pops in. And so that's how we can propagate the pressure inside something that's filled with liquid. Okay, and again, we have the cochleus unwrapped here, unfurled. And what we'll see is that inside there's something called the basilar membrane. There's a couple membranes, but this basilar membrane is going to be one of the keys. And so here's the tympanic membrane, the eardrum, here's the ossicles, here's this oval window and the round window at the bottom. So when a big pressure wave comes in, it bends the tympanic membrane, pushes the ossicles, that pushes the oval window in, that deflects the basilar membrane down and causes the round window to pop out a little bit. When there's a low pressure, so the big pressure wave comes, and then after that there's going to be low pressure, the tympanic membrane flexes out, that pulls the ossicles out, that pulls the oval window out, the round window sucks in, that causes the basilar membrane to flex up. Now, one of the things that's key here is that different parts of the basilar membrane are tuned to different frequencies. So those that are farther from the round and oval windows responds best to low frequencies. So if we put low frequency, this part of the basilar membrane vibrates the most. If we have this intermediate frequency, like 1000 hertz, it's sort of this middle part of the cochleus or the basilar membrane starts to vibrate. And if it's at a high frequency, like 10,000 hertz, it's the part that's very proximal to the oval and round window. And let me show you sort of a silly video, but once you see it, you can't unsee it. That shows this. Oops, let me back this up a little bit. So here's the cochleus. Sorry, cochlea. It's going to unwind here. Now we're going to look at the basilar membrane. So lower frequencies here, higher frequencies there. So low frequencies, higher frequencies. And so when there's a lot of frequencies coming in, the whole thing is sort of vibrating. Okay, this shows the same thing that we just saw. So lower frequencies cause this part of the basilar membrane to vibrate, higher frequencies on this side. And it's not as clean as that video would seem to indicate. Here's a more accurate representation. So 1600 hertz has a basilar membrane deflection distribution of something like that. By the time you get to lower frequencies, it looks something like this. So it's not, pinpoint precision. It's more spread out. Okay, so now we're going to talk how we go from a vibrating basilar membrane into a neural signal. So here's the basilar membrane, and there are these two chambers. So the oval window is connected to this one, so as it pushes in, it's going to cause deflection, and then the round window is connected to this bottom part. Here's the basilar membrane down here, and on top there's another membrane called the tectral membrane. In between here are some stereocilia cells. These are the cells that are going to detect a mechanical strain, and we'll see what that looks like here in a second. So yeah, so here's another view. So here's the basilar membrane, here's this tectral membrane. We have these stereocilia. So we have the inner hair cells, we have the outer hair cells. Here's the inner ones, here's the outer ones. It's the inner hair cells are the ones that are going to sense movement. The outer hair cells, as we'll see in a few minutes, actually serve as an amplifier for the system. Okay, now, so what's going to happen? Well, we have an anchor point for the basilar membrane, which is at this kind of orangey part, and we have a different anchor point for the tectral membrane. And notice that they're not aligned on top of each other, they're offset. So what happens then is when we have an upward force, so when there's low pressure, this deflects up, and it tends to bend the little hair cells towards the right-hand side here. When the membrane deflects down, that tends to pull these hair cells more towards the left. Right? So they're bending back and forth to the right and to the left as they go up and down. And so now we need to go from mechanical reception into an electrochemical signal. And how this works is that there are potassium channels that have a little linker. So there's a little tiny protein, and we'll see an electron micrograph of this here in a second, that connects the gating portion of the potassium channel to the next sort of cilia of the cell that's the next bit longer. And this one has another connector from its potassium channel to the next longer one. And so what happens is that as this bends, so as the tectral membrane slides to the right with respect to the basilar membrane, that pushes these cilia over, opens the potassium channels, causes a change in the membrane potential. When the basilar membrane deflects up, the hair cells tend to bend to the left, these close. So now as it goes back and forth, we're going to have ion flux versus no ion flux. Okay, and this is what they look like. So that diagram isn't too much of an exaggeration. So when we have these little bundles of the cilia, they are mechanically linked together. And there's one big one, the king, which is the kinocilium. And then it has rows of shorter ones underneath it. And each one of these has a little linker protein. So this goes to a potassium channel. Okay. And these inner hair cells are innervated by the dendrites of cells that are in a structure called the spiral ganglion. So these are spiral ganglion cells. And these come to form the auditory nerve that carries information out. So the dendrite is going to the inner hair cells, and then it sends its axons out, but the cell bodies are here. Because of the way the system works, the stereocilia can only detect bending in one direction. That's what this is showing. So here's a deflection. If it's going in this 0 degrees, which is kind of horizontal in this picture, you get depolarization and hyperpolarization. If you go 90 degrees, which would be kind of this way, you don't get any deflection. So it kind of makes sense. If you look at the spiral ganglion cells, then what we'll see is that as the membrane vibrates, we tend to get bursts of action potentials that correspond with this rising phase of the pressure wave that's coming in, or the deflection. Okay, so we have this auditory nerve that's going to come out. It's going to go to the medulla. Notice that it's going to cross the midline, just as in most of the sensory systems. So we saw this in vision, somatosensory. And note that, so because we have this crossing, it's not complete, and we'll see why in a second. We'll see this superior olivary nuclei and what it does. It's going to help localize sounds horizontally. Some of the fibers then go to this inferior colliculus. We have seen before that some of the visual information goes to a structure called the superior colliculus, which is right on top of this. And this is involved in moving the eye. So this is a way that you can control eyes based on auditory signals. It then goes to the medial geniculate nucleus, and this is an outcropping of the thalamus. So remember, in the visual systems, we had the lateral geniculate nucleus. This is the medial geniculate nucleus. And then it goes to the primary auditory cortex. And again, notice the left cochleus is going to go to the right auditory cortex. And this is likely why most people, when they answer the phone, they put it up to their right ear because the processing of speech is lateralized and is stronger on the left in most people. Oops. Okay. So how do we do sound source localization? And this is pretty cool. So here we have the left cochleus, here we have the right cochleus. And now imagine we have a source, so the nose is going to be somewhere here. So imagine we have a sound source that's on the left-hand side of the space. Well, the pressure waves propagate, at a finite speed, and so it's going to get to the right cochleus before it gets to the left cochleus. And what's going to happen is that the sound gets transduced, you generate action potentials, and these are going to propagate down, and they're going to come into this MSO, the medial superior olive. Same thing is occurring at the right cochlea. So we're going to generate action potentials. Now, see these neurons that are labeled A, B, C, D, and E. These are neurons in the MSO, and they only fire action potentials when they get input from the left ear and the right ear. So they serve as coincidence detectors. So if the action potential started here in the left ear before it started in the right, because the sound got here first, it's going to be able to propagate longer on this green path as compared to the purple path. And so the coincident activity is going to occur at this neuron E. And so we can actually read out the laterality of the sound source by which of these cells activated. And so that's how we do horizontal sound source localization. Vertical is quite a bit more difficult, and it's the reason that your pinna have this weird shape. It's because sounds that are above and below your head reflect off of this differently, and based on the sound qualities, you can estimate how high and low they are. And you can try a little experiment. If you fold your ear, your ear flaps over, and have someone snap their fingers either above your head or below your head and close your eyes, you can sound source localize when you don't fold them over, but when you have them folded over, you can't tell if your eyes are closed. So that's something you can try. Okay, now let's talk about the auditory cortex briefly. So primary auditory cortex, or A1, it's located on the superior bank of the temporal lobe. So here's the temporal lobe. It's inside a little bit. That's what this little scooper is showing. And note that it has an orderly representation. So when we saw the somatotopic mapping in the primary sensory cortex, we saw a retinotopic mapping in the visual cortex. Well, here we have a tonotopic mapping, meaning that frequencies that are similar are nearby each other. So here they're showing that by 500, 1,000, 2,000. So you have a smooth ordering of frequency sensitivity or tuning in the primary auditory cortex. Now there's a number of other higher order cortices that are collectively sometimes referred to as belt areas. In some mammals, there's up to 7 of these things that we're not really going to get into many of those. We are going to talk about one area, which is Wernicke's area, which again, here's the primary auditory cortex, this little blue part. Here's his Wernicke's area. And this is specialized specifically in humans in that it is dedicated to processing human speech. And damage to this area affects the ability of people to hear speech even from themselves. And what's interesting is that people who have damage here, they don't produce speech that's very understandable by other people, likely because they can't hear and perceive what they're saying. And so their speech is often characterized as word salad. And so, if you want to read this, you can. Oh, sure, go ahead. Any old thing you want. Oh, I would if I could. It kind of sounds like Dr. Seuss, but it gets, it's very hard to understand. And again, this is, oh no, this isn't Oliver Sacks. This is a different neurologist. Okay, now we saw that there's descending control of other sensory systems. We saw this in the somatosensory system, that in the spinal cord, there's descending control specifically of pain systems. There's also descending control of the interfusal muscle fibers, so you can turn the gain of stretch signals up and down by controlling this interfusal contraction. We also saw this in the visual system, that we perceive illusory contours and other things based on this top-down processing. Well, the same thing holds in the auditory system as well. And in this case, it actually serves as a mechanical amplifier. And what this tends to do, just like in some of the other systems, it tends to increase our dynamic range. And the dynamic range is, in this case, it's the distance between the highest and lowest stimulus magnitude. Again, in this case, it's loudness that can be faithfully encoded. And so if you can only encode a small amount, say you could only encode, sound volume is usually in decibels, say you can only do 20 decibels, but out in the world, it goes from, minus 40 decibels to plus 40 decibels. The way you can faithfully do that is by shifting the range that you want to hear. And so if you're in a very quiet place, you would shift your dynamic range. And what that means is you would increase the amplification. But if you're in a very loud place, then you want to turn down the amplification. So it's, it's kind of analogous to what the eye does with the pupil and with the biochemical amplification that occurs. But in this case, it's the outer hair cells. And so these are receiving descending control. And when they're hyperpolarized, they actually get longer. So here's a little dotted line. And when we say longer, it's just, it's not a whole lot. It's just a little bit. And when you depolarize them, they shrink a little bit. So they actually change size depending on their membrane potential. Okay, great. So how do you use that? Well, we talked about how the basilar membrane is deflecting up and down, and that's causing the tectoral membrane to shift back and forth, and that puts shear on the stereocilia. Well, what happens is, so here's a deflection upwards. If we depolarize the cell, the outer hair cells, that tends to shorten these. It actually pulls the basilar membrane up because we've now shrunken the length of these outer hair cells. When we have the deflection downwards, the cells hyperpolarize. And again, this is a descending command. So the command comes down to hyperpolarize the cells, that elongates them, and that actually pushes the basilar membrane away from the tectoral membrane. So, so if you look at the blue line, with the outer hair cells, we get an increase in the deflection up and down. And what this does is this can affect the fluid pressure and can drive the oval window in and out, which is connected to the ossicles, which is connected to the eardrum. And so this actually can cause vibrations and ringing of the eardrum. And this is referred to as autoacoustic emission. So if you go in with a very sensitive microphone, you can actually hear the system. So even when there's no input in here, this happens at sort of a natural frequency and it tends to resonate. But if you hear ringing in your ears, it's not usually because of that. It's usually because of tinnitus, which is the ringing sensation that follows trauma or other damage. So if you go to a really loud show where the bass is kicking and it's really cranked up, and you walk out and you kind of hear this ringing, right? That's a sign of damage, usually to the inner hair cells. You can also get this from infection or disease. Um, the human range is between 20 hertz and 20 kilohertz. That's for a kid. By the time you're in college, this is maybe 18, 19 kilohertz. In adults, it starts to get lower and lower. So every year, I would go to this in class and play these things, and I could hear or see the upper range of my hearing decreasing over the years. So this is why I don't know if this is still popular, but at one time it was popular in high school to set your cell phone to 19 or 20 kilohertz so that the teachers couldn't hear it, but it annoyed all the other teens in the room, like a dog whistle. Other animals have a much larger hearing range, so bats can hear up to 200,000 kilohertz, so at least 10 times as high as what humans can do. And hearing loss can be caused by a number of things: infection, ototoxic drugs. One of these being antibiotics, and this is the reason that you can't give some young children, antibiotics because it can damage the cochlea. Just normal aging and trauma. So if you use a jackhammer or work on jet airplanes and don't wear hearing protection, you end up accumulating damage more quickly. Okay, but the good news is if you completely trash your hair cells, you can regain some hearing with an implant. So this is a cochlear implant. So instead of the basilar membrane vibrating up and down to put shear on the hair cells, you can directly stimulate the spiral ganglion cells. All right, so here's the battery pack, here's the microphone, here's a wire, and then in the cochlea it kind of wraps up where these little gray things are. Those are stimulation sites. And because of the tonotopic mapping within the cochlea, you can stimulate, if you want high frequencies, you would stimulate some of these. If you want low frequencies, you can stimulate over there. So, it's not nearly as good as, the biology solution, but it's a way of reclaiming some lost hearing. Now, interestingly, there are auditory illusions. So last time when we talked about vision, we had these illusory contours where the brain just perceives like a triangle even where there isn't one, because it's an obvious explanation for the pattern that you see, right? That it's more likely that there's a white triangle on top of a few circles and another triangle, rather than all these things lined up precisely in that way. In the auditory world, there's something called the missing fundamental, and I'll give this a little bit of an explanation. So what you're going to hear, so most sounds, they have a fundamental frequency and they'll have harmonics. So whenever you hit a string, this also applies for brass or woodwinds or anything like that. But let's think of a string. So if you pluck the string of a guitar, one vibration mode will be the entire length, and that'll be the lowest frequency. But the string will also vibrate like this. So it'll vibrate at integers. So here's it divided in half. This is the first harmonic. Second harmonic is thirds, fourth, fifth, sixth, right? So it just integer multiples of that frequency, or divisors of that frequency. Okay, so now what we're going to do is we're going to play the fundamental note and just the first harmonic. And what you'll hear is that when you play, so we're going to hear 3 times. So we'll hear a sequence of increasing tones. The first one, or tone pitch. So it'll happen the first time we'll hear the fundamental frequencies and the harmonics. The second time we'll hear the harmonics only. And then the last time we'll hear the fundamental frequency only. So I'll play this the first time, which is the fundamental plus just the first harmonic. And so what that means is that it's the fundamental plus this first harmonic. So when you hear just the harmonic, it's going to sound higher pitched because it's the wavelength is half. So it's going to sound twice as high. So that sounds higher. That's the harmonic. Okay, so that's great. But now what happens if we play the fundamental plus the next 6 harmonics? So again, we're going to hear all of it together. Then we're going to hear just the harmonics. And then we're going to hear just the fundamental. Now see, this should be a little bit harder to discriminate. So the real comparison is between the first two, right? So the sound quality sounds a little bit different, but the overall pitch sounds the same. And the reason is when you play the harmonics, it's these things, and your brain understands that when it hears these harmonics, that the fundamental frequencies should be there, right? Because there's not really anything that would produce just these without that, or it would be very unnatural, right? You can do it with computers and synthesizers, but not with natural objects. And it turns out that pipe organ designers knew this hundreds of years ago. And when they wanted a really low note, some of them would have required pipes to be up to 70 feet long, and that would be too big for many spaces. So instead, what they did is they would hook up some of these low frequency keys to just play the harmonics of the fundamental that they wanted. And then you would just perceive it as a very low note. Okay, so this is a summary of the important concepts for the auditory system, and now we're going to shift over quickly to the vestibular system. So the vestibular system is information about head movement, and so there will be systems for rotational movement in 3 different axes, and this takes the same nomenclature as boats and planes. So we have pitch, yaw, and roll, and these are the corresponding axes. And there's also a system for acceleration, not just around these axes. So instead of these are the rotational axes, but there's also systems for translation. So if you're moving forwards or backwards. So how does this happen? Well, we have the structure called the labyrinth, which is the fluid component of which is continuous with the cochlea. And so this is why when you get an infection in your ear and gets clogged up, your balance gets wonky, as well as, being hard of hearing. So sometimes you're not hearing as well because of waxy buildup, but if you actually get an infection or the salinity gets off because of diet or disease or whatever, you can not only have hearing problems, but also balance problems. And what we'll see is that there's 2 otolith organs that sense linear acceleration, and then there's 3 what are called semicircular canals. That's these guys, and there's one kind of popping out of the page at you, which sense rotational acceleration around the 3 axes of the head, that yaw, pitch, and roll. Okay, so the labyrinth is continuous with the cochlea, and it has a similar structure. It also has these stereocilia that are in this sort of organization of tall to small, and they have these linker proteins. And so when something bends them, then potassium floods in. So similar as the cochlear stereocilia. The mechanical reception in the inner ear, it's very sensitive, and it's equivalent to detecting the thumb's width sway at the top of the Eiffel Tower. And this is really pretty big, and it sways more than that just on a not so windy day. So how can we detect such small movements in the presence of large forces like gravity, turning a car, things like that? And again, just like what we saw with the outer hair cells, it's adaptation. So pretty much all the sensory systems have systems to adapt based on how much of something is occurring. So essentially this means if you're on a boat all the time, you can turn down your sensitivity, whereas if you're very stationary, you can turn it up so you can sense very small movements. The way this works is very, very clever. There are these actin filaments. So these are, form sort of the skeleton inside of neurons. And there's a motor protein that essentially walks on this. And this is very much like the transport mechanisms that we saw that walk on the actin filaments. And there's essentially a spring that's connected to these potassium channels. And so what can happen is that if this walks up, it puts more tension on here, and then it's more sensitive. If it walks down, this is more slack, and then it takes a lot more bending to open these things. So again, it's a way of turning up and down the sensitivity. Okay, so first we're going to talk about the otolith organs. So you need some way of bending these stereocilia. They're not very big and heavy, so what nature has done is essentially grown a calcium carbonate crystal on top. So this serves as a weight. So when it's just upright, it's pressing straight down, and it doesn't cause the shear force that causes them to bend. It's only when the inner ear is tilted up or down that it causes those shear forces that causes the bending and causes the depolarization or hyperpolarization. So that's illustrated here. So when you're upright, not much is happening. If you tilt your head backwards, now the weights are pulled backwards, and you get shear and bending of the stereocilia. If you bend it forwards, you get the opposite. So here you're feeling the acceleration of gravity, but if you're in a car and you're going forwards, you need a way of transferring the momentum, which is essentially through the hair cells. So as you start going, the crystal is sitting still. So you're going forwards, this essentially bends backwards. Same thing when you hit the brakes, the car slows you down and then you're slowing the weight down. And so it's bending in the other direction. So that's how you can feel linear acceleration. And because it has this weight, then you get the signal the entire time it's occurring. Okay, so when you bend backwards, the whole time your head is bent backwards or you're accelerating, you get a signal. On the deceleration or bending forwards, you get the opposite signal. So you get the sensation of acceleration or deceleration. And you get that constantly. But how in the world do you separate out that linear acceleration from rotation around the head? It's not easy. And so what I want you to think about is if you had a hoop. So if you have a hula hoop and you fill it up with fluid, and so if I move, so imagine you've got a hula hoop and you pull the whole thing, you've got your hand on the top, you're not twisting it, you're just pulling it back and forth. The liquid it's just going to move along with the hoop. It's not going to slide inside the hoop with respect to the hoop. But now imagine it's filled up and now the center stays in the same place, but now you spin it like this. Now the outside is going to spin, the liquid's going to tend to stay where it was. And that's the basis of the semicircular canals. So you have these canals that are like hoops, they're filled with fluid, and at one point you have this structure called the cupula that sits here, and if the fluid inside isn't moving in lockstep with sort of the outer part of the semicircular canal, then it's going to push on this one way or the other. So imagine you start spinning it. So imagine that the head starts rotating in this orientation, so around this axis, like this. Well, everything's going to move, but the liquid isn't. It hasn't had the momentum transferred to it yet. Sorry, imagine it's going this way. That's what we're going to see. So if it starts moving this way, the liquid staying still, the liquid is going to put a force on to the cupula, right? Because the cupula is actually moving. This is part stationary. It gets bent. You have the stereocilia in there, and you get a signal out of it. Now, to sense in 3 independent dimensions or directions that you need for 3D space, you need 3 of these, and they have to be what's called orthogonal. So that means they're perpendicular in 3 spaces. So one of them is in this axis, one is in this axis, and one is along the axis that's coming straight in and out of the page right here. And so normally when you're moving your head, you're not in one of those axes, but you're in a combination of these axes. And so so if you're moving your head kind of diagonally around, you're activating each one of these to some extent, and your brain can decode that and know what direction and what speed you're accelerating. Okay, once the liquid inside that hoop is traveling at the same speed, then it's no longer going to push on that cupula. So what you get is a signal that's only for the acceleration, it goes up, or the deceleration, it goes down. When you're at a constant velocity, the signal doesn't change. So again, this is why if you're on like a merry-go-round or you're on a swing and someone twists you up and you start spinning, right? You start spinning, it goes up, but then you reach some, terminal spin velocity, it settles down, and now all of a sudden you stop it. Now you get the sensation of rotation in the opposite direction for a little while. Okay. Now, the vestibular system is hooked up to a number of structures. It's phylogenetically pretty old, meaning it evolved a long time ago, so it doesn't necessarily follow the same paths as the other sensory systems. We're just going to talk about one of the things it does. Don't worry about what the names of these structures are, but one of the very important things it does is to stabilize the eyes. So this is how you can fixate on something while you're moving around and walking. And this is very important for predators and prey animals so that you can still see even when you're moving. And a good example of this is that if you hold a sheet or something with text on it, your computer, and you shake your head around, you can still kind of read. And the reason is that the vestibular system gives a very rapid signal to control your eyes so that your eyes can maintain a fixation point. And if you want to see how profound that is, you can take your sheet of paper or something and just shake it with your hand, and you have no chance of reading it. And the reason is that you don't have any vestibular input in that case to move your eyes around. And it's even worse if you have someone else shake the piece of paper. Okay, so that's about it for the vestibular system. We covered that in kind of less detail than some of the other systems. Just know the main points.